This is a bit of a sideline project, and it's the first time I've actually spoken about it. So this has been really fun to write this. And I think some of the things I'm talking about are a journey that you've kind of already been on. So I'm actually, well, I'm hoping you'll find it interesting. You might be like, oh yeah, we've been there, and you might give me some feedback. So I'm your token cryo-EM talk, and I will talk about the data that goes into our structure, maybe a bit about that processing, but the where I want to make my points are really how we're looking at how the microscope collected that data. And this is a team of people that think and are passionate about this problem. But I guess the coding that makes this happen is between me and Dan Hatton. So we're the UK National Facility, giving access to UK and EU scientists, academics and people in industry to Titan cryoses to collect high-resolution EM data. That's 5 Titan cryoses, 1 exclusively for industry and 4 for academia. And this pulls in about 52 user sessions a month. And I love napkin calculations. So that's about 1 million images, cryo images per month, if you sort of do some extreme estimation. But I'll go on to maybe be a bit more precise about this. Anyway, it's a lot of data, and we collect movies. These are not necessarily individually blockbusters, but I want to stress that they're movies because you can start thinking about what we're actually collecting. 40 to 80 frames at about 1 angstrom per pixel. Kind of big, I guess. But yeah, what am I going to try to convey to you is that we're collecting a lot of data, and gosh, it's petabytes. Anyway, so what's the point? What are we doing? You probably all know, but single particle is about cryo cooling a specimen in ethane, in liquid ethane, so that we get vitreous ice, which is electron transparent, and then we can take images of that molecule captured in that state, and what we will have captured are the molecule in different poses. And when we illuminate these with a flood beam and an electron microscope, we effectively get little X-rays, little projections of the 3-dimensional structure, two-dimensional projections. We can pick those from our images. We look for the poses that are the same and then average them together. Because although our detectors did get pretty good recently, the signal to noise in here is bad. This is a low dose technique. So we average to bring that signal to noise to a better place. As I said, there'll be lots of different poses, and you can put those back together in Fourier space and then back project to a real space map. So these slides are beautiful, but they're actually a bit old. So you only see the secondary structure. And I guess you all know now, routinely, we can solve maps with sufficient resolution and quality to build atomically accurate models. So the aim of the single particle cryo-EM experiment is to collect enough high quality data to determine that map. And I guess from the facility hat on, maybe with everyone's hat on, in a time efficient manner. This is a bit of an oversimplification because users might come specifically wanting to achieve high resolution, but they might want to resolve heterogeneity because we can do that. We capture the molecule in a full range of motion if it has it. Or maybe the user wants to do both because they're difficult or their sample is difficult. Either way, let's just say high quality data, time efficient manner. And what does that look like on the microscope? It looks like a user spending some time on Google Earth, looking for a target, going from a sort of a global overview into this point where they can actually set up multiple shots to then take pictures within the shots to see their molecule. So users, they have their sample on this grid. It's a carbon foil with holes in. It looks like this. There are grid squares. Inside those grid squares there are holes. And then, as I said, we set up multiple shots in this. And quite a lot of time is spent doing this searching around, looking for your diamond light source and eBIC or apoferritin. And once you've got the images, I've talked a little bit about the processing that allows us to get to our structure, and we offer that as a pipeline to our users. I guess we will one day, and actually we're on the edge of going to 2 structures, perhaps in real time. But at the moment, I think the best thing is we have these pre-processing steps that are motion correction, so unblurring, looking at what the radiation damage is over the course of that exposure and trying to correct for it, doing some measurements on what actually happens to the images, because it's in electron microscopes, so all sorts of weird things happen that we measure and we correct. Doing particle picking, which used to be manual, but is now thankfully automatic, and that initial averaging into these 2-dimensional poses. So we offer that as a service, which should improve the speed at which people can look at their data when they get back home. But it does also mean if a user submits this pipeline, we get quite a lot of quality information on our microscopes and how they're running. So we really like it when people run our pipeline. It's quite a big task to run that thing, though. Well, I think it is, and I'd like to see if you think it is. So we run GPU nodes. A user might come, and if they choose to submit a pipeline, they get logged on to a 4 GPU node. They don't see any of this happening, it just happens. A movie takes about 5 seconds to correct. So for a reasonable data set size, that's actually 14 GPU hours to do just the motion correction. The other steps, this signal delocalization and picking is run on CPUs. It's about 6 CPU hours for a reasonable data set. And then remember, as I said, the movies contain lots of little images of the macromolecule. So in a reasonable data set, you might actually have 2.3 million pictures of the thing you're trying to solve the structure of. And then when you start averaging those, the war times go through the roof. So yeah, we put quite a bit of resource into this pipeline. And I can then tell you in 2022 on our cryo user program, and these are actually gross underestimates, and I'll come back to why. We collected 5.5 million movies. 2.5 million of those were put into our pipeline, and it's not quite a napkin calculation anymore, but if you go on from the stats in the previous slide, that's 10,000 GPU hours last year to process what went through our user program. So, I mean, I think it's exceptionally cool, but the point I'm trying to make is that EM, in a way, from where I sit, is a big data challenge in itself, each data set. And there's a whole lot of data reduction that needs to go on before you can use that data for structure determination. And my next point is, if you're doing that on a large scale, you need quite a few resources to put in. So we can do that as a facility within the diamond light source. I'm saying, come to eBIC. But for us, as I said, the nice thing is that pipeline, that thing that we're throwing resources into, does give us very objective measures of session success and sample quality. And we don't want to be big brother over people's samples necessarily, but if we can start looking at how well we do on a sample, depending on its quality, I think actually we can be pretty helpful to our users. So let me talk about the next part, which is keeping an eye on how things are going. And this is where we're starting to try to mine data of how the microscopes are performing. So where did this start? It started with me having a go at writing some Python code that produced some PDFs. So at the end of your experiment, you can walk away with a PDF that tells you, this is what things look like. This is how the microscope was set up, and this is how fast you were, etc. And a whole bunch of parameters that would allow you actually, if you wanted to come and process this data in a year, you could look at this and you'd be able to do that. So tapping into the idea of metadata capture to make these things useful in the future. This will go into eBIC, but it wasn't there yet, so I couldn't help but write it. But once you've got that, then I was like, wow, we can scan every single session that was run, and not just produce this report, but actually produce a database that describes an entire year. So this is 59 microscope parameters for each session, over 555 sessions. There's quite a few thousand data points, just literally telling us how the microscopes were used. And not everyone submits to our pipeline, so it's a bit more modest, but we also have a reasonable database describing what the samples look like. And we're not there yet. I think we're at the beginning of our journey in this, but I think now we have that ability to do that data mining. I think we can actually start to answer like how we are doing in terms of high quality collection and how we're doing in terms of time efficiency. And in speaking of time efficiency, let's see if we can do this quickly. So some fun insights are, is this big? Do you think this is big? This is like terabytes of data set of size of data set for each data set that comes with the microscope. That's 1.3 petabytes of raw movies. That's 66 billion electrons per angstrom squared detected in the entire year. Again, gross underestimation, and 500,000 microns of area imaged. If you were to image perfectly next to each other, you actually wouldn't image very much of a grid. So it looks like we're terribly unproductive. But this is 555 sessions. Each one at least can produce a cryo-EM structure. So big scale and kind of fun. Those users who are coming, they had 91,000 options of places to target on their grid. And they chose 10,000 of them. So it really shows that these people, our users, come and they know where to go, which is good. It means they're using the microscopes well. And we're doing well because of those squares that they targeted, we managed to collect a reasonable number of them. And then I've sort of told you about how everything from this point scales up, and then you get to 5.5 million movies, so on and so on. But an insight here from now actually having gone and looked at all of this stuff is it's 650 hours of human setting this stuff up and 6,500 hours of data collection. And I guess 10% is not that bad, but I really think there's room to improve this. And so yeah, we'd really like to make some tools that automate and reduce that setup time. There's a little insight. And this is just showing off, just really, that we know this stuff. We know how different parts of the microscope are used, what kind of doses hit the sample, how the user decided to focus the microscope. And you know, it's just showing off. And actually, we don't have great insights from this. And the data set is huge. And we would really like to correlate all of this stuff with experimental outcomes. But I'm just going to show you a couple of case studies. So the first is, and this is nice. There were sort of the setups on our microscopes. When you go to higher magnifications where you might want to target high resolution, the setups and the changes in configurations elsewhere that we run and have upgraded allow you to get pretty major speed improvements as you start zooming in mag. That's really, really cool. More data, brilliant. But we had to go and look at the numbers to actually be sure of this. It turns out that the sacrifice you pay for going to higher magnifications, potentially to get resolution, like there is a sacrifice. You don't actually collect as much area. So I mean, it all seems pretty obvious now. But this is great to see, because now we can go back to our users and say, well, okay, you want high resolution. You really do need to think about whether you're still going to be able to collect enough data to solve that structure at high resolution. We can give them some numbers now to tell them how much more data they're going to need to collect. And as a facility, I think we're going to be doing things to try and push these data rates higher up so that this becomes flat. So that's cool. Here's an amazing example of what someone did. And we can only spot this because we're at a database and can sort it and look for people who've done interesting things. Oh dear, no, this is amazing. This is someone taking 38 shots inside a hole of protein and using optical setup in the microscope to sort of, instead of moving the stage, which would take a long time and be quite unstable, change the optics in the microscope to literally like zip around and take lots and lots of images. This just wasn't possible some years ago. So for me is excellent. But we can look at two case studies, like two very different collection strategies where someone's gone traditional single shot per hole. They get a pretty decent rate of collection at a reasonable pixel size that this in theory would be able to get you to around 2 angstroms. Or someone else who's come with a different specimen, a different sort of support, a larger hole, they do multi-shots. The speed goes down, which is what I said to you on the last slide. Yeah, I mean, I'm sure they'll still get enough data to get their high resolution. They could go to around 1.2 angstroms with this. But just the things to sort of keep in mind that I don't think we could actually give objective advice on before. We can do the same sort of data mining on what comes out of the pipeline. And again, it's just showing off. But there's some interesting things in here. People seem to like to bring 200 and 350 angstrom size complexes to eBIC. Who knew? But okay, here's back to that case study and maybe an actual insight. If you look at the motion, how much the sample moves around as you're exposing it, which is a problem for us, except for now that we can correct this, but still you don't really want it there. If we look at the motion in these two data collection strategies, forgive the difference in scale, but you can see there was very little motion in this and quite a lot in that. And I suspect it actually has a lot to do with the support being different rather than anything optical. But what I'm trying to convey to you is now we can look at this stuff, we can track how the microscope was, we can track what the specimen was, and all of this analysis, and start to put this together into if you're bringing this grid, then you need this strategy, and this is what you might expect in your data. So I've said, I've summarized that each data set in itself is a big data challenge, and what I hopefully maybe done a little bit of convincing is that as eBIC, as the national facility, we're really well positioned to monitor over a large scale program configuration, user behavior, and performance. And I've shown you a little bit of just two examples of how a collection strategy might be determined by the experimental aim, but we need to keep in mind that that will influence the characteristics of the data, how the session runs, and how that data will be analyzed. And now that we have this, there's some simple things. We can feed back to users how they're doing. We can have a very nice quick way to look at what was successful and what was unsuccessful and try and avoid those unsuccessful cases. But then you start to think, and we're kind of going there, if you can do this in real time, then you can monitor a session, and then you can kind of look at the characteristics of that and look back at the database and make predictions as to how much microscope time might be required to run this sample that's currently on the microscope. And then we can think about unattended data collection and our versions of that. And this is the other talk that I'm not giving now, but we have this database. We can annotate all of our images, and what we're going to try and do is actually I described Google Earth, right, and a human kind of looking and knowing where to go. Why? Let's teach a neural network to make those decisions based on annotating those images with real data, with real outcomes. We'll put this into eBIC, so we'll look a bit more like sort of crystallography, and I think we're sort of stepping into defining what metadata is captured and what should be deposited as standard when raw data and maps go into databases. So I hope we can be productive in that area. And yeah, my big dream, well, just haven't had enough time yet. We've only just really got this thing working, but we'd like to look for trends, like hidden trends in configuration and sample and outcome, and understand something that we just haven't been able to see before because we haven't been able to access so much data describing these experiments on a big scale. And I'm honestly intimidated by that because I'm not a data scientist. I'd really welcome any thoughts or input from you on that ambition. So we're a big team because we are kind of 5 beamlines. It's a nice place to work. We're on Twitter. Do check us out. And if you're coming to Diamond, yeah, come say hi, and I'm happy to talk to you about your experiments. Do you want coffee, or would you like to ask me a question? Thanks. Coffee is a legitimate answer.
Any question?